All right, good morning, everybody. How's everybody doing? Good, some good. I like the grades. Cool. Thank you for obliging me with the name tense here. It's super helpful. I really like to make sure I know who everyone is in the class. It'll take me a little bit, but I'll get it soon. Very happy to be here in person today. Feels good on this snowy morning. I will record the lecture today. I'm recording it right now, and that will just get posted every day, usually within an hour or two after class. The link to that is in Blackboard. There's a YouTube channel for the course. I'm not going to send out an announcement or anything like that. You can just assume the class will be posted within a couple hours. So today's agenda looks like this. Oh, just kidding. Psych. Today's agenda looks like this. We're going to figure out how to use a computer. I'm going to get into embryology today. I've got like maybe half a class, maybe most of a class worth of lecture on embryology. We're going to go over it pretty quickly, but there's some fundamental ideas that are important as we start off our adventures in learning human anatomy. We'll then get into a little bit of histology, and this will be what will take us through pretty much the rest of the week and overlap with a lot of what we'll do in lab as well. And then we'll wrap things up. So in advance of class today, you had, you're signing up for visible body. I asked you to complete that ideological awareness survey if you hadn't already, and you had some initial readings in your textbook on embryology. Any questions on anything before we jump into our topics for today? Well, I keep looking over here, expecting this backpack to have like a little name tag. Can we make a name tag for the backpack? Does it have a name? No. Do they have a name? I guess she's the they. That's something for you to think about for next class. Coming up with a good name. Okay. Embryology is the study. I'm going to figure out where to stand here so I can get in the video and yet you can see the screen. Embryology is the study of the origin and early development of a single individual. So we're going to be focusing on human embryology because this is a human anatomy class, but this can apply to any multicellular organism. And you see here a scanning electron micrograph of an 8-cell human embryo. This is very early in development, obviously. We're going to be talking about this briefly, and the reason is it's important to understand how the body is shaped in these early stages so that we can understand its structure later in life. The class is not going to focus on the anatomy of embryos or fetuses really at all outside of today. Yet the foundation of the structure that we see today is going to form our bodies later in life. There you go. Make a little name tag if you don't mind. So let's get into it. So here are some reasons why we care about this. Is embryology not only helps us understand our current structure, but it helps us understand our evolutionary past. Looking at embryos, there's stages of development that are common across vertebrates. So for example, when all of you were wee little embryos, you had a tail, and that's neat because lots of vertebrates, indeed most vertebrates, have what's called a post-anal tail. So vertebrae extending beyond the anus, forming a tail. You all had it. You didn't have vertebrae at that point, but you had a tail-like structure, and that goes away later in development. But understanding those structures can help us understand how different organisms are related. Also, there are derivations of adult structures appear on lots of your standardized tests that some of you might take if you're looking to get into the medical field and things like that. So you just need to know this for those tests. In my opinion, not the best reason to know something, but important. Those of you going to med school, you want to do well on those tests. So we're going to cover it. And finally, exposure to what's called teratogens, it's a good vocabulary word, during development can lead to a variety of anomalies in later life. So for example, exposure to ethanol early in development can lead to what's called fetal alcohol syndrome, which is kind of the most common cause of mental retardation in the US, for example. So it's just one example, but there's many things that can happen early in development that affect your anatomy and physiology later in life. And so I just want to put a disclaimer here. The word teratogen actually comes from the Greek word for monster. And I think it's terrible that we use this word to describe people. And so I'm teaching you this word because it's the word that's used in the literature. But I think we need to get a new word that is not so offensive, in my opinion. So that's just my little disclaimer. I really don't like that word. You should know where it comes from and what it means. So there's two major stages of prenatal development. And we're going to break this down. We're going to talk mostly about the first stage, which is the embryonic period, the first 8 weeks. And then we'll talk about the fetal period, which is the subsequent 30 weeks or so. Human development takes about, from conception to birth, is about 38 weeks, give or take a bunch of weeks. Sometimes babies are born late, sometimes they come really early, so it can vary a lot. We're going to just look at it as about an average of 38 weeks. And you can see the general stages here. Fertilization happens. There's a week after conception. So in your book, actually, these pictures are to scale. They're obviously not to scale up here. It'd be scary if they were, but kind of cool. But in your book, they're actually to scale. So that's kind of cool. Check out this figure in your book. But you can see the general timeline here. What's important to note is this distinction. The embryonic period is only 8 weeks. The fetal period is 30 weeks. But we're going to spend most of our time talking about those first 8 weeks, because that's when really all the cool stuff happens and the body begins to take shape. So let's see, what do I want to say here? So here's a focus on the embryonic period. What happens here is what's called the germ layers form, and all of these organ systems, those structures are started. And this is especially the time when exposure to these teratogens can cause major damage and influence these developmental pathways. It's also when often the mother experiences peak nausea and vomiting during pregnancy. And we're not really sure why that is, just as an aside. There's different hypotheses for why women experience either aversions to certain foods or what's the word I'm looking for? What's the opposite of aversion? Quick, we're going to play the Anthony game. Affinity. Who said affinity? Yes, good one. Affinity for certain. So women can experience a really strong affinity or aversion to certain foods, pickles or chocolate or whatever during this period. They can also experience a great deal of nausea and vomiting. It pretty much sucks from what I understand, but we don't know exactly why that happens. Anyway, that's a little bit of aside. So what's important to note here is that there's 3 primary germ layers. And from those 3 layers, all of the subsequent body structures form. Now you'll notice that your textbook covers a lot more of these details of the blastocyst and the gastrule and all of these early stages. I'm going to skip over that in our class. You don't need to know that for the purposes of this class. In your reading for your textbook, notice that I told you, I gave you exactly which sections to focus on, and we skipped over those in favor of focusing on these germ layers, because that is really where these organs start to take shape. So here's a figure showing an embryo at 5 weeks post-conception. And what I want you to take away from this, and in subsequent slides, is this color scheme, specifically this yellow, this red, and this blue, right? And what happens here during our early formation is we have essentially our body plan. Do I get into this? I'm just looking at my next slide here. Our body plan is essentially a tube within a tube, right? Where our body is composed of an outer tube, and within that outer tube is an inner tube, which becomes our respiratory and digestive system. So we can see that in yellow here. The red is this kind of segmented outer tube. And so it's not as evident in humans how it's segmented, but you can see it, for example, in your ribs. Your ribs are a series of kind of not identical, but very similar structures that happen throughout the body. So you think about that happening here. And so these your muscles and everything is derived from this tissue. And then you have this what's called the dorsal hollow nerve tube. And that is what forms the brain and the spinal cord of the nervous system. I'm going to get into this in more detail. This is just an overview right now. Notice also there's something called the notochord in purple here. This is a structure that you have early in development, and in fact is the defining feature for all the organisms in our phylum. At some point in development, you have this notochord. Lots of organisms keep this notochord into adult life. So for example, if you've taken vertebrate anatomy, you've looked at lamprey, they have this notochord. We essentially lose ours during development, except parts of it end up becoming your intervertebral discs. So those discs of cartilage between your backbones, those are derived from this notochord tissue. So when someone talks about like a slip disc or something like that, that tissue, those discs are derived from this notochord. We don't have the actual whole cord, but it develops into the cartilage in between the vertebrae there. So this is the structure. This is an embryo at 5 weeks, as I said. You can see the tube within a tube structure here, the notochord. Notice that you have this inner tube that becomes the digestive and respiratory systems. And then we have this dorsal hollow nerve tube that becomes the central nervous system. And you can see a lot of similarities here with our friend the fish. And you can see especially how I mentioned that these section of the body is segmented here. The segmented outer tube, you can see that especially in fish, that muscular structure where it's these kind of repeated bands of tissue that develop very similarly. I'm going to pause here, see what questions you have right now. Go ahead, Danny.
You were talking about the germ layers previously. So
yep.
Did that have like, did you name those out, or were those like the inner tube?
I'm going to hold that question. I have a couple of slides coming up. I want to give the overview now. We're going to get back to the germ layers in a little more detail in just a second.
Okay. Last thing. So with when you're talking about, like, I think it was like the second or third slide about the teratogens, like what you mainly just have to know that that just leads to like a future problem in a.
Yeah, we're not going to get into any detail of like what exposure to what chemicals might cause what problems or anything like that. I just want you to know that it's a sensitive period because all of those organs are forming. And if you change a little thing when something is just forming, that's going to have a bigger effect down the line.
So the organs are just forming, so do that it like almost like halts it.
It can halt it or just change its developmental trajectory, right? Yeah. Good question. Any others? Cool. You don't need to know about fish, but I want to just show you this because it's important to see the similarity here in our body structures. We have this blue brain and spinal cord. That's that dorsal hollow nerve tube. We have these segmented outer tube here. And then we have this inner tube, which becomes the respiratory and digestive system. And we have a notochord here in the fish, which is also replaced by vertebrae in most fish, depending how you want to define fish, but that's a whole other thing that we won't get into in this class. You don't need to know about fish, but I want to show you the similarities because boom, look at same thing in people. We've got this dorsal hollow nerve tube with our brain and spinal cord. You see the segmented outer tube there, and then you see this inner tube in yellow. So pay attention to those colors, the blue, the red, and the yellow, because I'm going to go back now and go to Dan's question about the germ layers and show you where those layers come from. Question?
So the segmented outer tube just becomes like our skeletal muscles?
Yes, and I'm going to talk about that in detail, but for now, it's primarily your muscles and your bones and basically the tissues that aren't part of your digestive system or your central nervous system, kind of everything.
So bones other than the spinal cord?
Well, it's complicated. The spinal cord, remember, is nervous tissue, and the spinal column, the vertebrae, are skeletal tissue.
So it's even those bones.
Yes, yep, that's correct. Yep, good questions. Anything else? All right, so let's look at those germ layers. So here we see those three primary germ layers. Super important, you need to know the names of those three: the ectoderm, mesoderm, endoderm. Just as a note, remember that ecto means outside, meso or meso means middle essentially, and that endo means inside. So these prefixes will come up over and over again in class. I'm trying to think of a good way to remember. Ecto means, I keep thinking of like ectoplasm from what's the movie Ghostbusters, but don't remember it that way. Ecto, meso, and endo just means outer, middle, and inner. That'll come up over and over again in class. So make sure you know those. And derm just means like cell layer, right? And so what happens is your little embryo forms these three layers: the blue, the red, and the yellow. They're not really blue and red and yellow, just to be clear. We're just color coding them so you can see what happens to them. And then these germ layers fold up, and this is how you get this tube in a tube structure, right? This yellow tissue, the endoderm, becomes the digestive tract, the inner tube. The mesoderm, the red in the middle, becomes all your organs kind of in between. I'll show you that in a second. And then the blue is your ectoderm, which becomes your nervous system and also your skin and integumentary system. So your hair, your fingernails, your sweat glands, all that fun stuff. And so as those layers fold, they kind of fold up and then they kind of take this shape. So this is looking at it from a cross section. So this is a 24-day-old embryo, so 3.5 weeks in a sagittal section. So lengthwise, you'll learn this term sagittal in lab tomorrow. We'll get there. And you can see how those structures kind of fold on each other and how they take this shape, and then how that then becomes this adult structure. So pay attention to the colors here, and especially the inner, the middle, and the outer. Then this is a figure from your book. This shows you the derivation, what tissues come from those three layers. You don't need to know this in great detail, but what I, do want you to know is the ectoderm, the outer layer, becomes the epidermis of your skin, your hair, your nails, etc., as I mentioned. The brain and spinal cord. Now that's important because that's not really intuitive, that this outer layer becomes the brain and spinal cord, something we think of as inside our body. We're not going to get into the details of why that is, but just know that that's derived from ectoderm tissue, and then also some sensory structures and some other structures as well. But really, I want you to know that the epidermis, hair, nails, etc., and the brain and spinal cord come from the ectoderm. The endoderm becomes the lining of your digestive system, essentially. So all the tissue that lines your esophagus and your stomach, your intestines, your colon, etc. That's all derived from the endoderm. And then the mesoderm is basically everything else. You don't need to know the lateral plate mesoderm. I'm not asking you to know that stuff. I'm just asking you to know generally what structures are derived from that. And that includes your muscles, that includes the kidneys and your gonads. That includes connective tissue like bones, joints, ligaments, etc. Although joints not a tissue, it's a whole bunch of tissues, but cartilage, things like that. And then also your heart and your blood vessels. So if you know what comes from the endoderm and ectoderm, you can then pretty much guess what comes from the mesoderm. I will ask you questions about like, from what germ layer is your brain derived? You should say ectoderm. I'm not going to ask you really obscure things like from what germ layer is the fourth phalange on your, something like that. I want you to know general ideas here is what I'm interested in. Generally understanding this path, these developmental pathways. I've got a couple more slides on embryo. Let's pause here, see what questions you have, and we'll get to those.
So when it comes to the folding, you just have to know that like the ecto, meso, and endoderm, they fold on each other and sort of just like create that sort of lateral fold.
Yeah, I want you to know how they go from this bunch of flat tissue layers to this tube within a tube system.
Okay.
That's the takeaway message, and that's it.
So the folding of that the layers create a tube within a tube.
Exactly. Yep, precisely. So let's look at a cross section here. This is a transverse section of an embryo. So this is if you took an embryo, if the embryo is standing in the position I'm in and you took it and sliced it right about here, or actually, where is it? The neural tube, somewhere in the torso, basically. You can see this tube within a tube structure. Again, this is from your textbook, a little more detail here than you need to know, but I want to emphasize this tube within a tube. You have the endothelium. You have all this stuff in red the endoderm, which forms the endothelium of the digestive system in yellow. You have all this stuff in red is the mesoderm, and then you have the outer layer is the ectoderm. And notice that these nervous system cells here are also blue because they're derived from the ectoderm. And if we look at an adult, so this is looking at a transverse section of adult through like the middle of the abdomen, you can see basically the exact same pattern. Of course, those structures have changed and developed, but we can see the endoderm derived tissue, the mesoderm derived tissue, and then the ectoderm derived tissue. Notice that your limbs have basically the same structure, except there's no endoderm. It's just the mesoderm and the ectoderm. So the limbs kind of sprout off from the main body, and they don't have the endoderm tissue. So your muscle and bone are from the mesoderm, and your skin is from the ectoderm. What am I say here? Just looking to see what else I want to cover there. That's all I want to do. That's the big takeaway message here. And those figures are in your book. You can take a look at them in a little more detail. This is basically the same figure. I don't need to emphasize anything more in there. I'll just pause here. This has like the keys so you can see the color coding. Let me pause here. We're going to talk about the fetal stage in a second, but let me see what questions you have.
Why the ectoderm inside within more, not the outer layer. Is it the reason, like evolutionary wise, that it got smaller?
Here you mean the spinal cord?
Yes.
Evolutionary wise, that's an interesting question. I hadn't really thought about that. So your question is, if you look at an embryo, it's bigger relative to the rest of the body.
Yes.
That's an interesting question. So here's my best answer is that when you're developing, your nervous system is bigger relative to your body. So you think about babies got big old heads, right? Their brains don't grow as much into adulthood as the rest of their bodies do. And that's why if we looked at like the scale of a baby versus an adult, babies' heads are proportionally much, much bigger. And so the entire nervous system kind of develops at a different rate than other parts of the body. Why that's important evolutionarily, my guess is because you got to learn a lot when you're young. And so relatively, it's more important to have larger organ sizes. But that's honestly a guess. I'm not sure. Yeah, it's an interesting question. I hadn't thought about it like that. So you've got like proportionally, you've got more space filled up by those ectoderm derived nerve cells here compared to you do as an adult. Yeah, good question.
So I've seen that the spinal cord's like kind of inside the mesoderm. So like originally as it folds, does it get there by just like the mesoderm kind of growing and then like, I guess it kind of somewhere getting inside of it?
It's kind of this complicated folding and development pattern that we're not going to get into. There's details in your book, I would say.
Okay.
Yep, but you don't need to, for our purposes, we're not going to get that. You can take a whole class on, I mean, everything I've covered in the past 20 minutes, we could take an entire semester to really make sure we understand it. We're not going to do that. I'd encourage it. It's really fascinating stuff. But we're just not going to, it's not going to be the focus here. So that was human embryos from conception to 8 weeks in 24 minutes. Let's talk about the fetal period in about 24 seconds. Basically, all the stuff that forms during the embryo period gets bigger. There's other things that happen too developmentally, but the big takeaway is all of those organs, all of those systems form during the embryonic period. And then during the fetal period, they just grow. They get bigger, they get stronger, they gain functionality. But new systems aren't emerging. All of those things are there. They're just getting, taking shape and gaining the ability to do their job essentially. And so this can happen, this fetal period is defined as from about 9 weeks to birth. And notice that we can say 38 weeks or birth, because birth can happen sometimes as early as 22 weeks. And a baby can survive with intensive medical intervention. So with some medical technologies now, it's pretty amazing actually how teeny tiny some preemies can be born and make it, which is great, right? We like when babies live. That's all we're going to say about the fetal period. Stuff gets bigger and develops shape and functionality. Boom. Done. All right, we're going to do this. We're going to have a partner discussion. I can see in all of your eyes you wanted a partner discussion. So what I'd like you to do is work with a partner and fill in the blanks in the following sentence. And you can take notes on this as you're talking to your partner. In human development, the embryonic stage is characterized by stuff. Whereas during the fetal stage, stuff. So you've got 2 minutes. Grab a partner, no more than 3 if you can, if you need to, but I'd prefer just 2. And I want to have you just talk about this and come up with what words you'd put in those blanks. Go for it. If you need help with a partner, I'll come around. I'll just check, make sure everyone's got one. Yeah, you guys good? Yeah. 2, 3.
It's just like, so it's basically initiation and then it's kind of.
Yeah, differentiation.
So it's basically growing, you just.
Yeah, I feel like a lot more of the meat
Got about 30 seconds left.
first line.
It's still going. Oh, it's still going. I was hoping there's supposed to be, there it is. Look at that. All right, that was 2 minutes. Great, thank you. Next step here is I want you to go ahead and upload your response. Just a quick Google survey. It's okay, you work together on this. So if you both have the same answer, that's fine. But everyone should individually fill out a survey, please. Get the QR code there. Yep,
Make sure this is, okay. I'll-
yep, just do it in the survey. That's, yep. Just go ahead and put it in there.
If it's like a different setup, is that okay?
I kind of wanted it like this, so if you have something that works and answers this question, that'll probably be okay. I'm interested in understanding more than I'm in grammatical structure. Right? QR codes there. Everyone able? Let me know if you can't get it. I can help you. I'll use these a lot in class. I love QR code. I mean, the idea of it, but you know, we'll use them a lot in class for surveys and stuff. Is the internet bad in here? Okay, it shouldn't, I mean, it's just a Google form, so hopefully it won't take too long. Whoops. Has everyone got the code? It looks like it. You didn't get it. Okay. Oh, there we go. The timer is working. Once you're done, if you don't mind, just set your phones down and then I'll know, I'll be able to just kind of gauge when everyone is done.
No,
No, he thinks I'm hacking.
Casey, you got it okay? All right, looks like most everyone's got it.
You'll just have two case responses.
All right. By the way, now that we're halfway through class, how is this light? And generally it seems nicer when it's dimmer, but I didn't actually ask that question. Do we want more lights? Is these lights okay? Give me a thumbs up if this level of lighting is cool for you. It looks like most of you. Okay, cool. I just want to make sure that's kind of working. You can see your notes and the screen at the same time. All right, this is where 2 points is a class activity. We'll do lots of these throughout the semester. Sometimes you'll get up and move around and do different things. Today, just a little discussion to break up the lecture. That's all we're going to talk about with development. Half a lecture on it. We are going to transition now and talk about histology. This is going to take up the bulk of our work this week and into lab this week and maybe next week. And histology is the study of tissue. So in the first lecture, I talked about the organization of structures of the body from molecules up to organelles to cells, etc. Remember, tissues are a group of closely associated cells that perform related functions and are similar in structure. So they're cells that hang out and work together, right? And when tissues get together, that then forms organs. But it's important before we get into organs and all that to know what kind of tissues make up those organs. And there are 4 basic types of tissues. So I want to give you an overview here, and then we're going to talk about each of these in more detail, both in lecture and in lab. So the first is what's called epithelial tissue. Epi meaning on top of, the prefix EPI. And so epithelial tissue covers things. Connective tissue, as you might guess, connects things and especially provides support. Muscle tissue provides movement, and nervous tissue allows us to control and coordinate the actions of all of those tissues throughout the body. So there's only 4 tissue types. All of the cells in your body fit into one of these 4 categories. So here's what we'll do for histology. That was our introduction. I'm going to just walk through each of these 4 and give you a general overview of them. As I said, we'll talk about this in lab, and there's also a lot of reading associated with this. Your program for Wednesday has a pretty big chunk in your textbook to read, although you don't necessarily have to read it all by Wednesday. It's going to serve more as a guide and a reference as we work through these tissue types. We're going to spend this, I mentioned there's 4 tissue types. We'll probably spend about half of our time on epithelial tissues, about a third of our time on connective tissues, and then what's a half plus a third? 3/6 plus 2/6. So we have 1/6 left. And then maybe a little bit on muscle tissues and then a very little on nervous tissue. So bulk of our time on tissues is going to be on epithelial tissue. And that is because they're complicated and there's a lot of them. So here is a cross section of epithelial tissue that lines your intestine, right? And to show you the structure here, we've got this epithelium, that's epithelial tissues. We've got what's called a basement membrane, which is this kind of connective material between there. And then we have this connective tissue underneath. We're going to focus on epithelial tissue right now, but I just wanted to show you what it looks like in a place in the body where we find a lot of it, which is that endothelium that lines your gut. Remember, that's derived from which of the 3 germ layers? Endoderm. Good. Epithelial tissues are known, they regenerate well. So your skin is made up of epithelial tissue. So if your skin is damaged, it can regenerate rather quickly and rather efficiently. They serve to protect structures of the body. So your skin protects internal organs from the outside environment. They secrete things like sweat or oils. They absorb, they can absorb both like physical shock as well as in some cases nutrients. So for example, the epithelial tissue lining your intestine absorb nutrients so your body can use them. They also filter things out and they're involved in sensory structures as well. And so what we're going to focus on here is not that. Actually, I don't want to focus on that. Yeah, actually we will. Okay. So there's a couple characteristics of epithelial tissue that I want to focus on. The first is that there's very narrow extracellular space. So if you look right here, basically they're really tightly packed is the takeaway message. There's not a lot of what's called matrix between them. So if you're forming a protective barrier, it makes sense to be closely associated with the cell next to you, because if you're trying to form a barrier, but there's a bunch of space between you, you're going to be a crappy barrier. The second important characteristic is they have these tight cell junctions, and we don't need to know the types of these just yet, but there's different structures that allow these cells to be tightly connected to each other. And this can take the form of what's called a tight junction, an adhesive belt, a desmosome, or in some cases a gap junction, which allows nervous system signals to be transmitted quickly between cells. But the takeaway here is that there's tight junctions and connections between these cells, which of course is associated with the fact that they're packed closely together. We then, each cell has a basal region. And so they have what's called a basal region, which is kind of the part of the cell that's deep, that part of the cell that's kind of facing the inside of the body. And they have an apical region. That's the part of the cell that's facing the outside of the body. Now, to be clear, it's a little bit confusing on your skin. The apical region would be on the outside of the skin, and the basal region would be like the lower part, right? But remember, in an example in the endothelium, it's kind of the opposite, because the stuff that's in your stomach is technically outside of your body. So in this case, the apical region is like in contact with all the food that's in your stomach, let's say, whereas the basal region is the part that's kind of closer to your body tissues rather than that the food that you're digesting. I'll get you 1 second, Ethan. I wanted to do one more thing. Epithelial tissues are also commonly connected, or excuse me, associated with connective tissue. And so they, through this basement membrane, they're often adhered to connective tissue, which gives them an anchor and some structure, because in themselves, they might not be very strong to keep their shape, but by associating with this stronger connective tissue, they can be a more, I don't want to say structured barrier. Go ahead, Ethan.
So with the apical and basal region, would you kind of consider them like the apical always going to be part that faces in towards the center of the body, whereas the basal is part that faces out towards like the skin way?
It's going to depend if it's endothelial tissue. So if it's lining your digestive tract, then in that case, the apical region will be facing inward and the basal region outward, right? Because technically the outside of the body is what's inside your digestive tract. Whereas if it's a skin tissue, then it's going to be opposite. The apical will be facing the outside world and the basal region will be facing the inside of your body. Does that answer your question?
Kind of, I think so.
Yep. So its orientation will shift depending on whether it's on the outside of your body or if it's lining your digestive tract.
So in the inner tube of the tube,
Yep.
the apical will always face in.
Exactly.
And on the outer tube, the basal will always face out.
Basal will face in that case.
In, yes.
Yeah. So the apical is what it interacts with the outside world. And the outside world can be the air in this classroom, or it can be the breakfast burrito that you had this morning. Maybe some of you had breakfast. Anybody have a breakfast burrito? Get some chorizo in there, some green chilies, maybe? No? Wednesday, you got another chance. One more is the last. Yep, one more aspect of this is that these cells are often innervated. So we have parts of nervous system cells that provide sensory input from our epithelial cells, and also they are generally well supplied with blood vessels as well. So there's nerve tissue, and they're innervated and they're vascularized, meaning that there's blood tissue there to deliver oxygen, remove wastes, etc. So I just walked through those concepts on this diagram. This next slide here is basically just a list of those same things I said. So just to refresh your memory, characteristics of epithelial cells is they're separated by minimal extracellular material, which is known as like extracellular matrix. They're packed closely together, right? They're joined by special junctions that keep them packed together closely so they can form a barrier. Their apical surface is different from the basal surface. So they have this what's called polarity. So one side is different from the other. They're supported and nourished by connective tissue. They're avascular. Oh, okay. So I made a mistake here. Let me correct myself here. I apologize. I said that the epithelial tissues were well vascularized. That's not right. The connective tissue to which they're attached is often well vascularized, but the epithelial tissue itself is a very thin layer, so it doesn't need to be. So I apologize for that. I messed that up when I was talking about it before. So they're generally avascularized, meaning that the blood vessels don't go directly to those cells. Rather, they serve the connective tissue to which those epithelial cells are anchored. I'm going to pause here because I don't like when I screw up. I don't want to confuse anybody. Sure.
So like when you cut yourself and bleed, you've gone all the way through the epithelial tissue because it's not vascularized at all, the entire epithelial tissue.
Yeah, that's actually great. So if you get, where you get like a nick and just did it to myself, stupid. Don't do that. You get a scratch and it doesn't bleed. Generally, in that case, you're only damaging the epithelial tissue, right? And the center, it's avascularized, so there's no blood right there. When you cut yourself and you go deeper, you're going through the epidermis, the dermis, and then you're getting into that connective tissue, which has the blood vessels, which is vascularized, right? And that's when you're damaging those lower tissue layers that are vascularized. Yeah, that's actually a great way to say it.
But the whole skin falls into the epithelial tissue.
No, it's more complicated. We'll get to that next week. Yeah, the outer layer of the skin is epithelial tissue, but you have the epidermis, but you also have a dermal layer, and then you have connective tissue layer underlying that. So the skin is an organ composed of different tissue types, and this is one of those tissue types. That's great. I'm actually glad you asked that because that leads us into where we get to next week. Talk about fingernails and hair and all that fun stuff. Cool, good questions. Anything else? All right, a couple other points on epithelial tissues. Lost cells are quickly regenerated, as I mentioned, and it serves a number of diverse functions, especially protection, but also secretion. So sweat glands, oil glands, things like that. Absorption, so nutrients in the gut. Filtration, and we'll get to that. We talk about the kidneys, and also sensory organs are composed of epithelial cells. So what kind of epithelial cells are there? There are a whole bunch of them. And they're usually described by two words. The first word in the name describes the number of cell layers. So for example, we have simple or stratified epithelia, which, as you might guess, simple is just one layer, which you see there. And then stratified is multiple layers. And this bottom or basal layer is what's attached to that basement membrane. So there's simple and stratified. The second part of their name tells you what shape the cells take. And those are generally one of three types of shapes: squamous, which comes from the word for plate. Also, incidentally, the same root as the word squamate, which is the group of reptiles that includes snakes and lizards and amphisbaenians. Those of you that wanted to know that, but it basically means flat, or it means scale. And so they basically look like these kind of like a fried egg, right? It's got like the nucleus is a little bigger, and then this kind of flat area around it with the rest of the cell. We have cuboidal cells, which, as you might guess, are shaped like cubes. And we have columnar cells, which are like cubes but taller. So we can name our epithelial cells by naming whether they're simple or stratified, and then whether they're squamous, cuboidal, or columnar. And their shape can then indicate to you the different function that they play. And I'm going to talk about this, and then we'll wrap things up. I think I'll do one more slide here. Yep, that's actually perfect. So simple epithelia are often associated with diffusion and filtration. So because it's a thin layer, stuff can move through it. So when there's squamous, simple squamous epithelia is associated with diffusion and filtration, whereas simple cuboidal or simple columnar are more associated with secretion or absorption. And we'll see examples of this in your text. The reading for Wednesday gets into more details for each of these types. I'm just kind of giving an overview right now. For your squamous epithelia, you have stratified squamous, which is important for protection. So think about, you have a whole bunch of layers of these flat cells stacked on top of each other. Most often, or not most often, but very often, the outer layer of those are actually dead. So they're there, they're kind of hanging on, they're service protection, but they're not actually, the cells aren't receiving nutrients anymore. So your skin is a good example of that. You're constantly rejuvenating those lower layers of that stratified epithelia, and those outer layers are being shed. That's why something, I don't know, what's the number, like 75% of the dust that's floating around the room is human dead skin cells. It's I don't know, I'm making up the number, but it's a high percentage of dust in the air is like all of our shed stratified squamous cells floating around, which is neat to think about, right? Then you have stratified cuboidal or columnar. This doesn't happen very often, but they can serve to protect certain structures. We'll see examples of that in the lab. And then you have these special kind of cells called transitional, and these guys essentially can stretch a lot. So for example, in the bladder, you have a bunch of cells that they can be compact, but as your bladder gets more and more full with urine, they have to kind of stretch out. And we have these stratified cells that are called transitional epithelial cells that can help, the way they're structured helps you stretch out that tissue. Some of you might be feeling that right now. You're like, when's class going to end? My stratified transitional epithelial cells are distended fully. Yes, go ahead.
So what differentiates that from the squamous little alveoli?
The transitional cell structure. That's a great question. Let me show you that in lab tomorrow. How's that sound? We'll look at examples of what the structure are. Basically, the way they're shaped together basically allows them to stretch without getting pulled apart. Let me just show you a diagram tomorrow. Well, yeah, that's a great question. I just don't have that handy here. All right, let me skip to the end of the slides here. As I'm doing that, go ahead and think to yourself, what questions do I have right now? And I'll answer those questions. And then we'll do this. Questions? What time we got? Oh, we're just on time, 2 minutes. Okay, so as I mentioned, your class program for Wednesday has a lot of reading. You don't need to read it all for Wednesday, but it will be a useful reading to come back to over the next week. Also, there's a video posted there. I really like this Crash Course A and P video series. You guys have probably all seen these. They're 10 minutes long. They're really nice. Go ahead, Elijah.
Is there anything except
Hey guys, hold on 1 second, please. Don't pack up yet. It's hard for me to hear your questions. Go ahead, Elijah.
is there anything except the program we need to prepare for lab tomorrow?
Great question. And here we go. Your first lab is, it's like you read my mind that this is on the slide. First lab is tomorrow morning. Remember that the morning lab meets at 9 o'clock in the afternoon at 1:10. It's over in the other Science Center building, which is super confusing, but it's room 301, which is upstairs. It's my teaching lab. I love it over there. A couple of reminders for labs: wear closed-toed shoes in case anyone wanted to wear flip-flops tomorrow. Please don't. We will be using specimens and scalpels and things like that. It's just a safety precaution. Later in the semester, it'll become more relevant for you. If you have long hair, please pull it back with a hairband or something for lab. Again, because tomorrow, for example, will be, well, either tomorrow or next Tuesday, we'll be starting our cat dissections. You don't want to get your hair in the dead cat. That's gross. So pull your hair back, please. And then finally, because it's a lab, no food or drink in there. We do use different chemical reagents and stuff like that. We'll have a table outside. And the way I structure lab, I'll talk about it tomorrow, but it's very loose. You'll have work to do, but you can come and go as you need if you want to step out and get a drink. But I say that especially for the morning lab, students often like to bring their, their breakfast muffin or whatever to class sometimes, which I don't care about in lecture. That's great. But for lab, you can't do that because it's just for your own safety. You can't eat in there. You don't want to because there's like, formalin from dead cats and stuff, and that's kind of gross. So, Elijah, does that answer your questions about lab?
Yep.
Awesome. Let me know what questions you have. Otherwise, I'll see everyone tomorrow. Thank you so much. Dan, go ahead.